In this video, we will discuss about targeted protein degradation or TPD using nanoparticles. Because of the ongoing interest in this TPD to target, as it can target protein of interest with a generalized design. And it can use the cell's protein recycling system to degrade the targeted protein. So it can be used for different varieties of protein, and instead of using a labor specific targeting of a protein using the traditional approaches such as CAR design. So as a proof of concept, they designed their targeted nanoparticles to target a generally overexpressed gene proteins in the cancer, that is EGFR. So the design is shown in this panel A, where they took the antibodies against EGFR and make a conjugate with NHS-PEG-DSPE. This is FDA approved polymer, so they make a conjugate so that they won't face too much trouble in the clinical trials. So they make the conjugate with it and then add it and then mix it with this PLGA to form a TPD nanoparticles that can target EGFR. And they measured the size of this TPD nanoparticles, it's around 160 nanometer. And then they also check whether this PLGA is binding to the conjugate or not using Western blot, and they can see that the increase in weight in the PLGA or this TPD nanoparticles compared to the antibody alone. And after that, they check the targeted protein degradations, that is EGFR degradations in 10 nanomolar of this antibody concentrations, and they are able to see a decreased signals for the EGFR, and these things they have quantified also, and the decrease is statistically significant. Afterwards, they also check their EGFR signals, or how at which locations this EGFR has been degraded using microscope, and we can see that after like 24 hours they are able to see a decrease in the signals from the plasma membrane, indicating that the EGFR that expressed on the cell surface gets degraded. And in the supplemental figures, they have shown that once they remove this or once they replace the cell culture media, they are able to see again the overexpression against the overexpression of this EGFR. So indicating that this targeted TPD using based targeted protein degradations is a temporary process. It can be switched on and off based on the requirement. And these things, the microscopic images, are further validated using the confocal images, where the signals for the EGFR can be seen diminished when they use TPD nanoparticles compared to the other controls. And once validating that their EGFR is reduced, then they check the proliferations impact, and they are able to see that when they use their targeted protein degradations nanoparticles or EGFR targeted nanoparticles, they are able to see a reduced proliferations compared to the other conditions, compared to the controls. And the mechanisms they thought that would be working in this is that their EGFR targeted nanoparticles interact with the EGFR on the cell surface, and that further leads to the internalizations because of the because of this polymer, and once it is internalized, then it will go to targeted degradations, and these degradations could be through lysosomes or through phagocytosis. It depends on what kind of target this TPD nanoparticle has been designed. So apart from that, they have shown in the supplemental figures some of the results about when they replace the cell culture media which does not have these nanoparticles, and that I have just said, and I'm just repeating it. And in that case, they are able to see again expressions of this EGFR. So this shows the specificity of this approach. that can be used whenever it is needed. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.